Hello. So we are going to model the process of DNA replication today. And so you're going to get a kit with various materials, and you want to start by just setting things up, and then we're going to model the process. So first thing you're going to need is this strip right here. Make sure it's labeled sequence 1, replication, and transcription. And the first thing you're going to do is use this model to build a DNA sequence out of these little nucleotide pieces. So notice that this matches exactly the order that I have. And notice also that the two strands are anti-parallel, which means on this top strand the arrow is pointing that way, but on this bottom strand it's pointing the opposite way. So that's what we mean by anti-parallel. So your first task is going to be to build this complete DNA sequence. And then you're going to need to get some other things. It might help to have a little pile of each color nucleotide kind of sorted out that way. I've kind of pre-loaded mine to get it ready to make the video go a little bit smoother, but you can ignore these. And then you're going to need this structure, which contains the enzyme called helicase. And then you're also going to need two of these guys. These represent the enzyme DNA polymerase. And then finally, you will need a handful of these gray pieces, and these are going to represent RNA nucleotides. And so you can just kind of pull out a few of each of those. So what we're going to do is we're going to demonstrate how DNA is replicated. And so what happens is the DNA molecule is going to head into this enzyme called helicase. Helicase's job is to unwind the two strands. So at this point, I'm going to break the two strands apart, and that's relatively easy because these nucleotides are held together by hydrogen bonds. In this direction, they're held together by covalent bonds, and so that's going to be a stronger interaction than the ones between the two strands. So these hydrogen bonds are broken, and helicase essentially unwinds the two strands, and you can just kind of feed that on through. So eventually, an enzyme called primase is going to lay out an RNA primer. So since my first sequence here is G, G, C, G, I'm going to use the complementary RNA nucleotides to represent the primer. So complementary to G is C, complementary to G is another C, complementary to C is G, and complementary to G is another C. So these are RNA nucleotides, and this little sequence right here is called a primer. The reason that is necessary is because this enzyme, DNA polymerase, is unable to initiate DNA replication. It can only elongate an existing piece. In other words, it needs a nucleotide to attach something to. It can't just start from scratch. Primase can start from scratch. So at this point, DNA polymerase is going to start adding the appropriate nucleotide. So I'll just kind of start here, and T is complementary to A is complementary to T, C is complementary to G, A is complementary to T, C is complementary to G, and so on. Now, this strand is what we call the leading strand because it's sort of synthesized in order. And note that in each case, we're adding a nucleotide to the 3' end. Down here, this strand is called the lagging strand. And the problem with the lagging strand is it's kind of opposite. So I have to flip this upside down, our models, and then it kind of goes in this direction. You can see my nucleotide lining up. There's a gross hair in there. Lining up here. And so this has to be sequenced in a little bit different way. So the first thing that happens is another primer is laid down by the enzyme primase. And so that is going to, let me just see here, 1, 2, 3, 4. So a primase enzyme is going to lay out a short little primer here, and I shouldn't have DNA polymerase here. This is primase laying out a short primer. And then once again, DNA polymerase is going to come in and add complementary nucleotides. So note that we're synthesizing this in the opposite direction as the other one because again, we can only add nucleotides to that 3' end. So A is binded to T, C is bound to G, G is bound to C, and so on. And now we're at the end. And so you can kind of see the problem here. So as this unwinds a little bit more, or continues to be unzipped by helicase, the lagging strand has no problem. We can add the T, we can add the G, we can add the A, and we can just keep right on going here, moving this, or sequencing this in a continuous manner in the 5' to 3' direction. But down here, of course, since it's in the opposite direction, we need to once again create another little primer so we can continue going. So here's my primer, and I believe my primer goes here. So primase is going to lay down these RNA nucleotides to create a new primer. And then DNA polymerase, another DNA polymerase, is going to come in and it's going to add complementary nucleotides and continue. So at this point, we're sort of synthesizing this lagging strand discontinuously as a series of fragments called Okazaki fragments. And so this one's going to keep getting built again in the opposite direction as the leading strand, but still in that 5' to 3' direction. And then something interesting happens. Eventually it catches up to the primer that we laid down before. And so what DNA polymerase is actually able to do is it can actually remove the RNA primer and replace it with a new nucleotide. So it's removing and replacing, removing, replacing, removing. And then one last little issue here. It turns out that DNA polymerase is not able to connect this nucleotide to the next Okazaki fragment. So we need a new enzyme is needed called DNA ligase, and DNA ligase is what's able to seal that final covalent bond and complete the strand. And so you can imagine if we kept this process going, the leading strand will keep being built in this direction, 5' to 3'. The lagging strand, we're going to have to introduce another primer and then build in the opposite direction. Another primer, build in the opposite direction. Another primer, build in the opposite direction. So leading strand is continuous, lagging strand is discontinuous.